Hi there, my name is David Lawson and I have two roles at the moment actually, both at the University of Bristol. So I'm a senior lecturer in the School of Biological Sciences and I'm also the academic director for Inclusion, Belonging and Community. So in terms of what I do and what my work looks like, I'm exploring questions around flowering plants and the interactions that they have with their pollinators, and mainly I'm looking at why flowering plants and flowers in particular are as complex as they are, and I do this with bumblebees as study organisms. So in terms of what my work involves, I work in a lab where there are colonies of bumblebees, and then we use these bumblebees as a model organism to answer questions about plants and their flowers. So oftentimes that might be making up different kinds of flowers and then seeing if the bees themselves can differentiate between them, if they can learn different aspects of them to answer different questions like, why are flowers as complex as they are? Why do flowers communicate across so many different sensory channels? For instance, flowers are kind of broadcasting on all sorts of different sensory channels, like there's the visual aspects of them, there's the scent, the olfactory aspects of them, there's electrostatic components of them, there's texture components of them, there's gustatory, which is kind of taste components of them, there's architectural components. So there are all of these different ways where flowers are kind of communicating or informing with their potential visitors, potential pollinators, or floral antagonists, which are visitors that might have interactions which aren't beneficial to the flower. So my research is kind of looking into different aspects of these displays and finding out what these particular display components do, what do they mean for a visiting pollinator, and how does that benefit the plant, essentially. So for me, a typical day might be, well, every day is different really. So on some days I might be teaching, some days I might be doing more research that's related, that I can do on a computer, for instance. But for those days where I'm actively doing that plant pollinator research in the lab, that might be coming down to the lab and first you gotta set the bees up and get them going. So just like us, they might be a little bit lazy and lax in the morning, so feed them up. So we'll have kind of fake flowers in the arena which I'll pop some sucrose solution into, and then generally that starts to get a few of the bees going, and they'll start kind of drinking from the flowers in the flight arena and then going back into their hive box where they'll regurgitate that sucrose and then get right back out there. Once the bees have got going, then it's time to start setting up for an experiment. So that might be getting the artificial flowers ready to go, setting them up. If we were doing something like an experiment on spatial fragrance patterns, so the kind of distribution of scent across the floral surface, so maybe different scents in the middle compared to the outside, or a different arrangement of the scent. So there might be some time kind of pipetting the different scents, which would be mixes of essential oils, into these artificial flowers to then go into the arena, set that up in the arena, and then one by one trying to let the bees into the arena. And once you've set that up all and you've tried to get all the bees back into the hive box, you can set that up, that arena, clean it down, set it up with your artificial flowers, and then start your experiments, which the bees will, a single bee will come out that you've controlled using that gate system, and you can start recording what flowers they're landing on, have they made a correct or incorrect choice? Like, do they land on one of the non-rewarding flowers and leave before drinking, which would be a correct choice? Or do they land on one of the correct ones and start drinking, which would be another correct choice? And seeing how many correct versus incorrect choices. And we'd use that to see if they are learning. Is that proportion of correct choices increasing over time? And that can tell us, okay, so they are picking up on something and they're using that to inform their decisions. So what makes this work so exciting for me is that we get to ask these kinds of questions and try and find out the hidden components of the strategies that flowering plants are using to get outcomes that are beneficial for them. So what kind of strategies have they evolved to change the behavior of their visitors, their pollinators, their antagonists, to get a maximum benefit? So I think it's really fascinating that because the plants can't tell us themselves, we do the scientific work to slowly uncover all of the fascinating strategies that they've evolved to live their lives and to reproduce and to attract pollinators. And I think that's just really fascinating, just diving into that hidden world, which helps us understand how are pollinators kind of perceiving the world, how do they interact with flowers, why do they do that, and as well on the flower side, why are they incorporating this part of their display, or why are they investing in all of these different display components rather than just one. So I think it's just fascinating to be working in this world that's full of mystery and using the scientific method to uncover that mystery. I think is wonderful. In terms of the qualifications that I needed to get to where I am today into this particular role, so I did a undergraduate degree in animal biology at the University of Gloucestershire, and I was lucky enough to get a first for that. And I didn't actually do a master's, which often a lot of people do, before starting my PhD. So firstly, I spent many years working in hospitality and in pizza restaurants and behind bars and doing all sorts of odd jobs. So after my university degree, I didn't really know what I wanted to do. So I fled to the jungle and I did a conservation internship. So I saved up money in one of those pizza restaurants and used that money to go to the Amazon in Ecuador, and I did a conservation internship there where I was doing all sorts of different kind of scientific data collection things in the forest, a lot of biodiversity surveys, and it was there that I thought, oh yeah, I really love this. This is what I'm all about. I know that I want to be continuing to do this. And when I came back to the UK, I applied for a PhD at the University of Bristol and was lucky enough to get that PhD. So that was kind of the trajectory that I went through. My advice for future biologists would be to keep your mind open with the different subjects and topic areas that you're engaging with. A lot of students that I see, for instance, they're not initially interested in plants, but then the more they find out about them, the more they realize, plants are really interesting. They've got just as much going on as the animals do, but they just have the limitation that they can't move around as quickly. So keep your mind open with different topic areas. There might be some that on the surface you think, I don't think I'm going to be interested in that, or I think that's going to be a little bit too difficult for me. Try it out, or read up some stuff on it, because you might find it much more fascinating than you initially thought.